So far in this unit, we've looked at DNA and RNA. In LO3, we're looking at how we're going to make proteins, this first step. Our first step is going to be copying DNA using RNA, transcription. And then in LO4, we'll look at taking that one step further and using the RNA to make a protein from it called translation. A couple reminders that are really important that you understand before we can go into transcription. DNA is housed in the nucleus and it's not allowed to leave. It's so important that we don't want to take it out and accidentally screw it up. RNA, however, can make a copy and then go out of the nucleus into the cytoplasm, and so it can actually leave. RNA copies a gene, not all of DNA, just a small section of it in the process called transcription. In translation, the RNA will be read because it's a code to help make a protein. Before I get into the steps of how transcription actually works, let's watch an animation. Here we have two strands of DNA because DNA is double-stranded. You can see it, the A's and T's paired, G's and C's. It's just like what you did at the end of LO1. You had a sequence of DNA and you had to write the other sequence for it. Now we're going to make a copy of one of the genes. So first, we have to actually unzip our DNA. There's a molecule called RNA polymerase that's going to work with some other molecules to open up DNA so we can start making a copy of a gene. Now note, it's not copying the whole strand, it's just copying a small portion of it, the gene. That gene of DNA, that small section that we want the code for, is copied by this molecule called mRNA. And remember, RNA is different from DNA. It's only one strand. It has U's instead of T's, all those things that we talked about in LO2. DNA will close up, all of the molecules involved will leave, and then we can take our mRNA, send it out to the cytoplasm, and do whatever we need to do with it in order to make a protein. In order to cover what happens here, I'm going to go through the steps. That animation was very simplistic. And then at the end, you're going to see a video that shows this process and how awesome it is, how intricate it is, and that will sum it all up. So, first, you need to realize that we are still in the same language as DNA. Transcription is simply going from nucleic acid language. However, we have a different dialect. Instead of using T, now we're using U because this is RNA. It'd be like if I was talking to you in an accent. You can still understand what I'm saying because we're still talking in the same language. However, there's a dialect difference. There's a slight difference in how I sound. It'd be like DNA could be regular English that we're used to, and then RNA could be British English, where, again, still the same language, just a slight difference in the way that things are pronounced. Same idea in DNA versus RNA. Transcription happens in the nucleus because that's where DNA is. So DNA, just like you saw in the animation, is going to be unzipped, and then we'll copy it, and then we can send that RNA out. The major players in transcription, we have the gene from DNA. RNA polymerase was that big molecule that you saw in the animation. We have mRNA, and in the picture here, the big green blob is RNA polymerase, and as it's unzipping DNA, the RNA strand, that copy, is coming out. And then we also have free nucleotides. So where do the A's and U's and G's and C's come from that make up RNA? Well, you have extra bases floating around inside your nucleus, and so a part of RNA polymerase's job is to grab those free nucleotides and put them where they belong. What actually happens in this process? First, we find where that gene is located. You have 6 feet of DNA, and we only need a little piece of it. So we have very complex machinery that allows us to find where that specific gene is. That big honkin' molecule, RNA polymerase, unzips DNA, opens it up. RNA polymerase grabs free nucleotides floating around in the nucleus and starts making a complementary copy of your DNA. The nucleotides are matched to their bases, A with U because this is RNA, G with C. When mRNA codes the whole gene, so once we hit that sequence that we need, this whole process stops. The pieces all fall apart, go along their way. The mRNA goes out of the nucleus through a pore into the cytoplasm where we can do the next step. where we can read it and actually take that code and make a protein out of it. Again, what's the point of this whole process? We need to make a copy of DNA so we can use the instructions to put together amino acids. Amino acids make a protein, and proteins are needed for just about everything inside an organism. A final video clip is going to show this whole process in real time. It has molecular animations of what this actually looks like. Very cool. So you can see the whole process as it occurs. More detail than you need to know, but still an excellent video. And after you're done with that video, then check in the note packet for what's next and see me if you have any questions.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.